the last talk in this session. I hope you'd find it interesting. Um, okay, I'll talk about my PhD work, the molecular probes targeting the GLUT5 transporter protein. I'll just give a brief introduction about the breast cancer. Currently, is the leading cause of cancer-related death and the most diagnosed cancer among women in 2020, with around 2.3 million new cases globally. However, over the last decade, there has been significant decline in the breast cancer mortality rate, and this is maybe due to the advancement in the tumor diagnostic tools used for early detection and different treatment options that become available for all cancer patients. The key to successful management and therapy of breast cancer is screening and timely detection, and currently there are several traditional and modern diagnostic techniques, including mammography, ultrasonography, MRI, biopsy, and the molecular imaging technique, which is the scope of our work today. This technique includes in vivo visualization, measurement, and diagnosis of the breast tumor. It gives very good specificity and contrast between normal and breast cancer tissues. In this technique, an imaging agent, maybe a radioactive atom or a dye, is injected inside the patient's body, and it's selectively uptaken by the breast tumor tissues. Then the movement and location of the substance is traced by different detection methods. And generally, these imaging agents are mainly targeted molecules labeled with either fluorescent dye, so it can be detected by fluorescence microscopy, or radioactive atoms, so it can be seen by PET technique. As we all know, all the cancer cells have these 8 characteristic hallmarks, in addition to 4 additional hallmarks that were reported in the beginning of 2022, in which the rapid cell division and reprogramming of energy metabolism are 2 critical hallmarks. And due to their abnormal metabolic profile, tumor cells demand an excessive supply of energy, which is fueled mainly by hexoses. So cancer cells, they are characterized by excessive fructose and glucose uptake to supply this needed energy in a phenomena which was described as Warburg effect. So to supply this high hexose consumption, tumor cells overexpress the facilitative hexose transporter proteins, which known as GLUTs, in comparison to the normal cells. And in clinical practice, the upregulation of this GLUTs protein in cancer cells was used as a tool for breast cancer diagnosis. And a very well-known example is a radioactive F18 labeled 2-fluoroglucose, which is currently the most widely used PET radiotracer for breast cancer detection. Now I'll talk about our target protein, the GLUTs. They are transmembrane transporters that mediate the hexose passaging across the cell membrane from the extracellular space to inside of the cell, and vice versa. As we can see, the glucose, fructose, or any hexose, it binds to the empty carrier, which undergo conformational change to release this bound hexose to inside of the cell, and then the protein shifts back to its original conformation for another hexose binding. And to date, there are 14 known isoforms of GLUTs from 1 to 14, and they are classified into 3 major classes according to the amino acid sequence, structural similarity, substrate preference, and tissue specificity. And actually, in class 2, they mainly mediate the transport of fructose and glucose. With the exception of GLUT5, which is our target protein, it mainly transport or have high affinity to fructose transport with little or no affinity to glucose. And normally, this GLUT5 protein is expressed in the small intestine, skeletal muscle, and the kidney tissues. So what is the significance of this GLUT5? As I mentioned before, all GLUTs protein were found to be overexpressed in tumor cells to fuel the increased hexose consumption, and in particular cases, cancer cells switch their metabolic needs and increase the consumption of fructose rather than glucose as an alternative source of energy. So the principal fructose transporter, GLUT5, was found to be overexpressed in different cancer types, including acute myeloid leukemia, renal cell carcinoma, lung adenocarcinoma, pancreatic and colon cancer, and our issue today, the breast cancer. Moreover, despite the overexpression of the GLUT5 in breast cancer cells, it was found to be tightly regulated in the normal healthy breast cells. So, in conclusion, GLUT5 was found to be a crucial biomarker, and it can be used as a portal for targeted delivery of anti-cancer drugs and a portal for tracer probes for molecular imaging of breast tumor. So our main goal was to develop probes consist of targeted molecules. It may be fructose or its analog, and generally this moiety should show high affinity to GLUT5, and then it should be attached to either radioisotope for PET, so it can be detected by PET technique, or attached to a fluorophore, so it can be seen by fluorescence detectors. Now I'll just talk a little bit more about the fructose and their analogs. It was found that fructose itself, after the removal of the C2 hemiacetyl hydroxyl group at C2, it affords this C2 symmetric fructose analog. It's called 2,5-anhydromannitol, and actually it displays better affinity to GLUT5 in comparison to the natural substrate fructose itself. And then modification and this compound was done, and the modification at C1 hydroxyl group of 2,5-anhydromannitol compound was explored, and it was found that different C1 modified analogs were reported as high affinity GLUT5 substrates. So the next step was to look at the C3 modification for their GLUT5 tolerability, which was the main idea of our work. Then our work was inspired by this study. It was conducted by former students in Dr. West group in 2017, and they aimed at exploration of C3 positions of 2,5-anhydromannitol derivatives. And in this study, they did synthesis of different compounds, including these 4 compounds which were installed by mainly hydrogen bond donor moieties at C3, including this NBD fluorophore, 2,4-dinitrophenyl acetamide and trifluoroacetamide, and 2 compounds incorporated by mainly hydrogen bond acceptor at C3, including the fluoride, the fluorine and azide group. Then the affinity of these compounds was measured through the inhibition of reference compound, which was C14 fructose uptake inside EMT6 breast cancer cell lines that overexpress GLUT5. Okay. Then it was found that the 4 compounds installed mainly with hydrogen bond donors, they showed a high affinity to the fructose transporter GLUT5. And on the other hand, the 2 compounds installed with mainly hydrogen bond acceptor, they didn't show any affinity for GLUT5, but instead they showed affinity for the main glucose transporter GLUT1. So from this study, it was concluded that the presence of hydrogen bond donor moiety at C3 was crucial for GLUT5 recognition. So the main idea of our work was to design new series of C3 modified to 5-anhydromannitol derivatives bearing different hydrogen bond donor functionalities at C3 and payloads of different sizes. So me and Natasha Rana, my partner in this project in University of Alberta, we did synthesis of 17 compounds, including these 3 aniline derivatives. They are installed with the electron withdrawing group to increase the hydrogen bond donor capability of this amino group. Also, these 4 sulfonamide compounds, including this densyl fluorescent probe, 3 thiourease compounds, 4 urease, and 3 amides. Then I'll show the biological evaluation results of these proposed compounds. Actually, we did 2 types of assays: the confocal imaging of EMT6 cells with with our synthesized fluorescent probes, and the in vitro evaluation of C3 modified derivatives as GLUT5 ligands in comparison to 2 reference compounds. In the first in vitro assay, it was a radio-labeled assay against the reference compound F18-6-FDF. Actually, we selected this compound as our reference compound because it was studied before, and it showed high GLUT5 uptake into both murine EMT6 and human MCF7 cells. Also, it showed accumulation inside the EMT6 tumor in mouse in vivo. Okay, so in this assay, we used the potent and well-established F18 labeled fructose derivatives, and it's the competitive inhibition of its uptake inside the EMT6 cells was measured against our synthesized compounds, and the IC50 values were measured. 8 of our final compounds were selected for this radio-labeled assay by Dr. Frank Wust's lab in the Department of Oncology, Cross Cancer Institute in the University of Alberta, and these 8 compounds were submitted to this radio-labeled assay. And here is the concentration-dependent inhibition curves for all these 8 compounds, in which the compound number 8A, the aniline derivatives, it inhibited. Okay, at first I forgot to mention that we did this in vitro assay against the cold probe 6-FDF and the natural substrate fructose. Okay, if we can look at the results, we can see that the aniline derivatives 8A, it displayed 10-fold better inhibition of F18,6-FDF uptake with IC50 value of 1.1 millimolar. Also, compound 9D installed with the bulky densyl fluorescent group at C3 through this sulfonamide linker. Also, it displayed a significant inhibitory activity. And finally, compound 19, this imide compound, also it displayed significant inhibition. And high affinity of GLUT5 of these 2 compounds may indicate that GLUT5 can tolerate this bulky densyl group or the aromatic group attached through a long teaser at its binding pocket, as we will see in the molecular modeling studies. The next type of assay was a novel fluorescent-based assay. Actually, the reference compound used here wasn't the ready labeled one, but it was our own available fluorescent probe 6-NBD fructose. And actually, it was a more suitable alternative to the ready labeled F18,6-FDF, as there is no need for radiochemical synthesis, and it can be easily measured by our readily available fluorescent plate reader in our labs. Again, why specifically we use 6-NBD fructose as a reference compound? It was studied before in 2017, and it displayed significant inhibition of C14 fructose uptake in EMT6 breast cancer cells. So, inspired by its high affinity to GLUT5, it was used as a reference compound in this assay. And again, the competitive inhibition of its uptake inside the EMT6 breast cancer cells was measured in the presence of our synthesized compounds, and the IC50 values were measured. In this assay, in addition to the 8 compounds tested before in the previous assay, all of our compounds were submitted, except, of course, of the 2 fluorescent probes. And here is again the concentration-dependent inhibition curves for all the synthesized compounds. We can see that the 3 aniline derivatives, they exhibited very strong inhibition of 6-NBD fructose uptake with low IC50 values. And actually, this compound 8A, its result was consistent with the previous result obtained from the previous assay, which showed which was reported as a potent inhibitor of the F18,6-FDF uptake. For the other class, the sulfonamide, the 2 compounds 9B and 9C showed significant inhibition, and also the thiourea compound 10B. Also from the urea class, 3 compounds show significant inhibition, and again from the amide compounds, compound 19 with the long aminoxy teaser, it showed significant inhibition, which was consistent with the previous assay. The next type of biological evaluation results was a qualitative confocal imaging, and mainly it was performed to determine if the 2 synthesized fluorescent probes 9D and 10C can be translocated across the cell membrane to inside of the cell, and which GLUT is involved in the transport. I'll just show the images of probe 9D as an example. We can see that incubation of probe 9D with the EMT6 cells, there is bright fluorescence was observed from the intracellular space, indicating that the compound can be transported to inside of the cell, not just binds to the surface. And incubation of 9D probe with glucose, there is little or no reduction in the fluorescence signal, but incubation of 9D with fructose, there is substantial reduction in the fluorescence intensity, which indicates that this probe can be fluxed across the cell membrane through the principal fructose transporter GLUT5. Then I'll move to the molecular modeling studies. Molecular docking and dynamic simulation studies were performed in just 3 compounds on fructose and the 2 potent inhibitors, 8A and 19, and it was mainly performed to predict their binding mode and interpret their high inhibitory potency. And here is a snapshot of the MD run of fructose inside the binding pocket of GLUT5. It exhibited hydrogen bonds with glutamine 167, serine 392, asparagine 294, and tyrosine 32. And actually, these amino acids was consistent with the reported essential amino acid residues for fructose binding, in which the glutamine 167 was essential for specificity of fructose to GLUT5. Then we did this MD simulation to compound number 8, as I told before, which exhibited the strongest GLUT5 binding. Again, it exhibited the essential hydrogen bonds with glutamine 167 and asparagine 294. In addition, the 2-nitro group formed hydrogen bonds additionally with histidine 419 and glycine 163. Similarly, the potent compound number 18, it exhibited these essential hydrogen bonds. So in conclusion, to explore the GLUT5 binding site requirements, 5 series of C3 modified to 5-anhydromannitol derivatives linked to different hydrogen bond donor functionalities were designed and evaluated for their GLUT5 affinity through 2 inhibition assays. And these 3 compounds, 8A, 19, and 9D, they displayed significant inhibition of the uptake of the 2 reference compounds. And in addition, this fluorescent probe 9D, through the confocal imaging, it shows that it can be entered inside the cell through GLUT5 protein. And finally, in the molecular modeling results, it showed that the ability of the 2 potent compounds to capture the essential hydrogen bonding interaction within the binding pocket in comparison to the natural substrate fructose. In conclusion, or finally, I want to thank my supervisors, Dr. Khalid Abu Zaid, Dr. Rabah Ahmed, Dr. Zina, and Dr. Nermeen in Shams University, and Professor Dr. West from University of Alberta, and Natasha Rana, which was the partner of this project there. And I want to thank you for listening and your attention. Thank you.